Welcome to Biology for Bastards, teaching biology in the most profane way you've ever seen or heard. I'm your host, John Doty. Thanks for listening. This season, we are going through the AP Biology curriculum. We are all the way on Chapter 20. Holy shit. That's almost the home stretch. So, this chapter, Biotechnology, is going to be a quick one because it's pretty straightforward. It's just a bunch of definitions, essentially. So, as always, there is the PowerPoint up on our new website, biologyforbastards.com. So, get on there and you can follow along. But we're just going to dive right in and start talking this biotechnology, which, as we know, because this is an AP class and we're grown-ass adults and all that shit, that bio's life, technology is shit, so this is life shit, essentially. So we're really talking about, if we want to use the textbook definition of biotechnology and not leave it as life shit, it is the process of manipulating organisms or their components for the purpose of making useful products. This uses the idea of genetic engineering, where we're manipulating genes, we're manipulating genomes to get desired outcomes. There's a bunch of different ways that this can happen. We could have recombinant DNA, where we're making our own DNA, essentially. We use that for like making insulin, where we insert the human gene into E. coli to get insulin made. We have gene cloning, where we just clone genes in the lab, and all this shit can happen to it as a result of having multiple copies. We can do our experiments there. We have restriction enzymes, which are coming from bacteria, and what they do is they cut DNA at specific locations called restriction sites, and it creates restriction fragments. And what these restriction fragments are just chunks of DNA that are formed whenever the restriction enzymes find whatever DNA sequence that they cut at, whatever that restriction site is, and wherever that shows up, it makes a cut. So if its restriction site is looking for where it goes GAATTC, and then the bottom strand would be CTTAAG. So it's palindromic. So if it's looking for that sequence, wherever you have a GAATTC in the DNA, it will make a cut, no matter where it shows up. Okay, that's how restriction enzymes work. And then we have DNA ligase that we'll use to make some recombinant DNA. We have vectors, viruses that we use for our benefit with all this genetic engineering shit. But it's pretty straightforward, all this stuff. So in the PowerPoint up on the website, there's a really good picture of how restriction enzymes work, where it makes this cut where there's some single-stranded overhangs, and we call those having sticky ends, because the single-stranded ends do not like to be single-stranded, so they want to stick onto something. So we can make these multiple cuts using these restriction enzymes, and then DNA ligase, will join them together to make a recombinant DNA. Like I said, recombinant DNA is where we have DNA from different sources, like that human insulin gene, into the E. coli. We can use something similar if we're doing any sort of cloning. We can insert genes into plasmids and then take those plasmids and insert those into bacterial cells, and then la-di-frickin-da, you have this gene in place, and then we let it copy itself over and over and over and over and over and over and over again, and now we have this shit ton of bacteria doing what the fuck we want, using weird ass genes that it normally doesn't have. So like I keep going back to insulin, that's how you can do it. Right? Now, there's other techniques when it comes to genetic engineering. We have transformation, which we have talked about way back when we talked the Hershey-Chase experiment. No, that's not transformation. Transformation was Griffith and Avery. My bad. Hershey Chase, that was... Fuck, I can't remember the word. Anyways, not important. I was just trying to be, overarching and combining stuff and reviewing, and it's just not working today. So, fuck it. Moving on. But transformation is where bacteria takes up a plasmid. So it's a plasmid that we made. We put the gene that we wanted in, and then we insert it, where we allow the bacteria to insert it into its DNA. We have PCR, which is polymerase chain reaction. And this is kind of along the same thing of, you let the bacterium take up the DNA and then you let it replicate over and over, and you end up with a shit ton of bacteria that have this gene of interest in it. PCR kind of eliminates the bacterial middleman, and it just amplifies a piece of DNA without using cells. You break it open, you take the little piece of DNA, you separate it, you allow it to anneal, and then extend, and then you have 2 strands of DNA, and then you repeat that process, where now you have 4, and you repeat the process, and you have 8, and then you repeat the process, you have 16, 32, 60, and you just keep repeating this process over and over and over again, and you end up with a shit ton of DNA out of a little bit of DNA. And then you can do various things with that. I worked in a lab during college, and we would do PCR all the time. We'd take a little sample of DNA from mice, we'd run a PCR to get a shit ton of DNA from them, and then we'd run the next thing, which is called a gel electrophoresis. And what those do is it separates the DNA based on size and charge. So DNA, the saying is, it runs the red, so it moves towards the positive pole because it's a slightly negative molecule. And this is what you might imagine when you think of like forensics and everything, and they put the little thing up and you see all the little lines and all that shit. That's a gel electrophoresis. And the one that looks cool but is a little tougher to understand is a microarray assay. So this is where you're studying the expression of a bunch of different genes at once. And essentially what you do is you isolate the RNA, mRNA. Then you make some DNA from it using reverse transcription, and it's labeled with these fluorescent nucleotides. And then we let shit happen. You put a different gene in each spot on this really tiny, or not a really tiny plate, a plate with really tiny wells. And then as you do stuff, it will fluoresce depending on gene expression. You can get all sorts of pretty colors. And the more colorful, the more fluorescence, the more active the gene is, the darker it is, the less active the gene, so on and so forth. We can clone shit using biotechnology in the process of nuclear transplantation. This is how we clone the first animals, Dolly the sheep, and all these other things. Now you take the nucleus of an egg and you remove it, and you replace it with the nucleus of a body cell. And then you grow that in culture a little bit, and then you implant it into another organism, and then it grows, and you give birth to a clone. So that's kind of weird, but it's kind of cool. The problem with cloning is that it's kind of fucked up. Usually has some problems. It usually has these epigenetic factors that have changed over the organism's life, and now you're implanting essentially an adult cell into an embryo. It's not even a child or a fetus, it's an embryo. So things that it's picked up during its life are now being implanted into this developing series of cells, and as a result you can end up with some unexpected consequences, let's put it that way, where they're typically not as healthy, they don't live as long, there's just some sort of issues with these cloned organisms. So that's cloning. Stem cells is another field or another thing in biotechnology where we have talked, I think, about stem cells a little bit. But basically, these little fuckers can just reproduce indefinitely and turn in all of these other types of cells. And depending on which type of stem cell we have, whether we have the zygote, which can become any fucking cell, embryonic stem cells that can become most cells, or adult stem cells that can become, a lot, but not nearly as many as the embryonic or the zygote. Should probably say totipotent, pluripotent, multipotent. Zygote being able to become everything is totipotent. Embryonic stem cells being able to become many things is pluripotent. And then the adult stem cells, which are the shittiest of stem cells, are multipotent, meaning they can just become a few things, but you can induce them back into their pluripotent stage where they can become more types of cells, or more different types of cells, if that makes any fucking sense. It doesn't sound like it does, but whatever. Now, using stem cells, we can use this as treatment. If we have an individual that has something fucked up with them, some sort of tissue damage or something like that, we can withdraw stem cells. We can reprogram them using the stuff that we've talked about so far, and basically force these stem cells to make healthy cells that are damaged in the individual. Then we can return these cells to the individual where they will replace or repair the damaged tissue, and kind of reversing the damage of whatever may have been a heart attack or whatever the fuck. We can do kind of along those lines is the idea of gene therapy, where we take the RNA version of the healthy or the normal allele, stick that in a retrovirus, let the retrovirus infect bone marrow that we took from the patient, kind of like I just said with the stem cells and everything. And then once that viral DNA gets inside the chromosome, because that's what retroviruses do, we can inject those engineered cells back into the patient, back into the bone marrow, and effectively we have repaired that gene. So it's got a fucked up gene, we go in, we give it a proper copy of it, then we let the virus infect it, and then we replace the fucked up cells with these kind of healthy cells that we created. We also have DNA fingerprinting for figuring out who the father is or who killed whom, all this stuff. The new way they do that is with these short tandem repeats, where the non-coding DNA, because most of our DNA doesn't code for shit, but it's got these sequences that are really short, like 2 to 5 bases, that are just repeated. And every person has a different number of repeats at various locations in their genome. And using that, we can figure out that pattern. We can match up one person's DNA with a sample DNA. So whether we're trying to figure out the father or trying to figure out who the murderer is, we can use these short tandem repeats for that. The old way of doing it was using RFLIPs, or restriction fragment length polymorphisms. And with this, you would have to go in with restriction enzymes, chop up DNA, and it's kind of the same idea, where each person would have a different number of fragments produced using these restriction enzymes. And then you'd run the gel, and you'd get this fingerprint. You would get this series of lines, series of bands on the gel that would be unique to an individual. But that's the old way. Now we use the short tandem repeats. You can get by with a lot less of a sample. RFLIPs, you needed like a quarter size sample of blood. With the short tandem repeats, you need about 20 cells. So that's a fuck ton less. So, and then lastly, GMOs, they're good. You insert a DNA into the genome or combine some stuff. You can put vitamins into rice. That's the big example. You can put shit into like corn, so you don't need pesticides. So the bugs eat it and they just die, so you don't have to spray. Everybody's afraid of GMOs because they're fucking idiots. GMOs are good for you. So yay GMOs. The most commonly genetically modified foods are the stuff that you use the most. Stuff like dairy products, tomatoes, potatoes, rice, cotton, soy, corn, all that shit. And that pretty much brings us to the end. I told you it was going to be short. It was going to be quick. There's not a lot of difficult concepts. We're getting into some easier shit now. I know the last one or two of them ago were pretty short. This one's pretty short, so hopefully everybody's cool with that, because I don't give a fuck if you're not. And that brings us to the end. So, if you haven't checked out our new website, do that. biologyforbastards.com. That's pretty awesome. I am trying to figure out how to sell merchandise. Shirts, mugs, hats, shit like that. I'm working on figuring that out. So that will be cool if I can figure that out. But until then, just keep spreading the word. Rate, review. If you haven't subscribed, I don't know what the fuck you're doing. Subscribe already. But tell people about it. Get the word out. There will be a second season. We've got about a dozen more episodes this season. But there definitely will be a second season. I'm still figuring out how the fuck that's going to look, whether it's another class or just some random episodes, kind of like our bonus ones. I've been tossing that idea around with some people. So we'll figure out what the fuck to do when that time comes. But we can focus on this season for now. I'm getting a little sappy here because this was a short episode, but I don't give a shit. I never expected this to be what it is now, and it's only getting better with every episode. I'm hoping to get more people so I can get better equipment, not have dogs barking in the background all the time, which I appreciate you guys putting up with. But I'll just kind of wrap up before I get too mushy. So, I'm just kidding. I'm fine. So this has been Biology for Bastards, teaching biology in the most profane way you've ever seen or heard. Our intro and outro music is Feeling Good by Purple Planet Music. We are on Facebook and Instagram and Twitter. All three of those sites, we are at Bio for Bastards. Our website is biologyforbastards.com. Check us out. Give us a follow on all of those social medias. And until next time, thanks for listening.